Autism-linked immune problems are caused by maternal gut bacteria. Children diagnosed with autism spectrum disorders, ASD, often also suffer from inflammation of the gut, resulting in gastrointestinal issues. A new study based on mouse models of autism, led by scientists at Harvard Medical School and the Massachusetts Institute of Technology, uncovers underlying mechanisms that trigger ASD-linked gut inflammation and GI symptoms. The researchers show exposure to maternal inflammation in the womb increases susceptibility to bacteria-induced gut inflammation in the offspring later in life. The study zeroes in on interleukin-17A, a cytokine that is heightened in the mother following an infection during pregnancy. The authors show IL-17A not only alters brain development in the fetus, but it also changes the mother's gut microbiome that in turn affects the offspring's immune system by altering the chromatin landscape in a subset of immune cells called CD4-positive T cells. This change in immune development primes the offspring for inflammatory attacks of the gut after birth. The new findings are reported in an article titled "Maternal Gut Bacteria Drive Intestinal Inflammation in Offspring with Neurodevelopmental Disorders by Altering the Chromatin Landscape of CD4 plus T Cells," published in the journal Immunity on December 7. The current paper focuses on how increased IL-17A can affect immune development as well. Choi said, "One of the striking findings we made is that offspring's immune cells remember what happened in the maternal womb, and this information gets transferred by the mother's gut bacteria. The study suggests maternal immune activation, MIA, due to infection during pregnancy is a common environmental driver for neurodevelopmental and immunological symptoms in individuals with ASD. There has been no mechanistic understanding of why patients with a neurodevelopmental disorder have a dysregulated immune system," said Hupp. Commenting on the study design, Choi said, "We used a multi-pronged approach encompassing immunology, germ-free work, neuroscience, and behavioral biology. Although neurodevelopmental abnormalities in the offspring in response to maternal MIA clearly develop prenatally, the time point when immune dysfunction starts in the offspring exposed to maternal MIA in the womb is not clear. To identify the time window when the immune system in the offspring is affected, the researchers switched mouse pups at birth so that pups born to moms with MIA were reared by normal moms, and pups born to normal moms were reared by MIA moms. From these cross-fostering experiments, the authors inferred that while neurodevelopment is altered before birth, immune dysfunction in the offspring is altered postnatally. The authors then probed how mouse moms with MIA affect the immune development of their pups postnatally. Earlier studies have shown that the maternal microbiome in the gut can influence the offspring's immune development." Choi added, "We hope to identify detailed mechanisms by which maternal gut bacteria can affect offspring's immune cell function by changing the way their DNA structure is organized. Emerging connections between inflammation and neurodegenerative diseases, such as Alzheimer's, may also warrant further study in the light of the current findings that indicate maternal infection increases inflammation in offspring postnatally," Choi said.